So let's look now at some interesting difficulty of NMR spectroscopy of proteins, which is the broadening of peaks, and how we might go about avoiding that problem. I'm going to use as an example human protein disulfide isomerase, PDI. PDI has four domains. The catalytic domains containing disulfide points are the A and the A prime. The primary binding site is in the domain B prime, and you can imagine PDI as being dissected into four domain fragments: the A, B, B prime, and A prime. Different sizes, they've got different weights in the slide. You also have this X section, which is just a sequence of 20 amino acid linkers between two of the domains, and the full length of the PDI has also a C fragment, a C terminal tail. So the whole molecule is, the whole protein is 57 kilodaltons. So this is the spectrum, the NMR spectrum of the A domain. And you can see, the peaks are relatively sharp. The spectrum is complicated because it's a fragment of a protein, but it can be interpreted. This is the NMR spectrum of the domains E and B together, 24 kilodaltons. And as you can see, well, the spectrum starts losing a bit the sharpness of the peaks. This gets even worse when you add the third domain, and when you do the NMR, proton NMR spectrum of the whole protein minus the C terminal, you can see the peaks are very broad. So this is a problem when you are analyzing larger protein fragments. The peaks get broader. You can still tell it is folded. You can still see that methyl section, but the peaks are definitely much broader, and you are lacking information. Why does it happen? Because as the protein weight increases, as the molecular weight increases, the protein it tumbles slower in solution. So that creates broader peaks, because you are seeing an average of all those conformations in solution, which results in peak broadening. The same problem can be found in 2D NMR. So for example, this is the nitrogen 15 HSQC NMR, and you've got the A domain. Can you see the peaks? The signals are larger when you add the B domain, even larger when you add the B prime, and the full protein, as you can see, the signals are very broad and cannot be interpreted. So what can we do about it? Well, one thing we can do about it is make them tumble quicker by providing kinetic energy, which increases that movement. For example, by heating the sample, it increases the tumbling rate, and it lowers the viscosity of the buffer solution, so it makes it easier for the protein to tumble. The problem with heating a protein is that you increase the risk of denaturization. So for example, this is an NMR data from a bacterial protein at 30 degrees. At 40 degrees, and you can see the peaks are getting narrower, and at 50 degrees. So yes, the peaks are getting narrower with increasing temperature, with the same time and scan parameters, but you risk, you start getting peaks which show an evidence of hydrolysis in the spectra at higher temperatures. Another thing that can be done is deuteration. Just to reduce the amount of hydrogen you have to worry. You can, instead of labeling carbon 13 and nitrogen 15 only, which you were already doing for normal 2D NMR, you also label the hydrogens. You use deuterium instead of hydrogen. and that reduces NMR broadening of the peaks due to NMR relaxation. So this is a TROSI spectrum, nitrogen 15 TROSI spectrum. TROSI stands for Transverse Relaxation Optimized Spectroscopy, and this is PVA at 37 degrees. And the peaks, TROSI is similar to HSQC, but the peaks are narrower. And there is less overlap between the different signals, so you can see the details easier. So what happens when you swap hydrogen 1 for hydrogen 2, hydrogen for deuterium in a proton NMR spectrum for PVI? Well, the red spectrum is the normal spectrum with hydrogen H1, and the blue one is a proton NMR spectrum of deuterated PVI of PVI enriched with deuterium. And you can see that, well, deuteration is only 60% efficient, and only non-labile hydrogens that are not involving exchanges are converted and will remain as deuterium. When there are exchanges with the hydrogens in the solvent, those will not get converted. So we are looking at a deuteration of non-active, non-labile hydrogens. And well, the NH is the same because you have that interaction, that interchange, that acid-base exchange between the hydrogen attached to the nitrogen in the peptide bond and the protons in the solvent. The aromatic section, if you compare with the spectrum of the non-deuterated protein, is reduced. You are losing those peaks from those aromatic those aromatic hydrogens. And the aliphatic area, it also becomes simpler because again those hydrogens are not labile. They are not going to be involving in any exchange with the acid-base exchange with the solvent. And as you can see, that area of the spectrum is also much simpler. So this is the end of this presentation, this series of videos on the biological applications of NMR. It's a great technique. It can be used to monitor small and large changes in proteins and peptides with proton NMR. With nitrogen 15 NMR, you can get information about the amino acids in the structure, which amino acids are specifically involved in particular reactions and interactions. And as you said, you can look at folding and unfolding events, you can look at conformational changes, you can look at oxidation status, reduction potential, etc. So basically it is a very powerful technique which allows us both to identify the structure of proteins, and also allows us to follow quantitatively metabolites and metabolic flux in metabolic processes involved in protein production. So just a list of all the people that provided some of the NMRs that I show in the video, and a special thanks to Dr. Liebern for providing the examples in which this video is based. So I've turned on my camera again to say goodbye to all of you. I hope this was useful. Please engage with the links to the literature I've provided on Blackboard on research papers related to the use of NMR in the study of proteins form and function. I hope it was useful. If you have any questions, please drop me an email, or we can discuss it in one of the sessions we're going to have together. And thank you very much for watching.